Hello and welcome to our channel AllGrow. In today's video, we will be going to discuss the topic aseptic transfer. Aseptic transfer refers to the process of transferring microbiological material such as culture or sample in a manner that prevents contamination. It is a crucial technique used in various scientific and medical fields, including microbiology and biotechnology. Aseptic transfer is employed to maintain the purity and integrity of the transferred material, ensuring accurate, reliable result in experiment, research, and clinical diagnostics. The primary objective of aseptic transfer is to prevent the introduction of unwanted microorganisms into a sterile environment or the cross-contamination between different samples. Microorganisms can easily contaminate culture or sample, leading to inaccurate result and compromising the validity of experiment or analysis. Aseptic technique helps to minimize the risk of contamination and maintain the sterility of the working environment. To perform an aseptic transfer, several principles and practices are followed. First, sterile environment. The transfer is carried out in a controlled and sterile environment, such as laminar flow hood and biosafety component. These specialized workstations provide a filtered airflow that reduces the presence of airborne contaminant. Second, personal protective equipment (PPE). Proper PPE including gloves, lab coat and mask is to prevent contamination from the operation. The PPE act as a physical barrier between the operator and the material being transferred. Third, disinfection. The working area and the surface of equipment and tools are disinfected with appropriate disinfectant before and after each transfer. This step helps to eliminate any potential contaminants present on the surface. Fourth, flame sterilization. Instruments such as incubating loops and needle are sterilized using an open flame. The metal part of instrument is heated until it becomes red hot, effectively killing any microorganisms present. Fifth, aseptic technique. Aseptic technique involves careful handling of material to minimize the risk of contamination. This includes avoiding slashing or aerial formation, keeping container closed except during the actual transfer, minimizing the time that samples are exposed to the environment. Sixth, proper sealing. Transfer materials such as culture plate or tubes are immediately sealed after the transfer to prevent contamination from the surrounding environment. Seventh, single-use tools. Whenever possible, disposable tools such as pipettes and syringes are used to eliminate the risk of cross-contamination between samples. Proper waste disposal. Contaminated material and waste are disposed of correctly to prevent the spread of microorganisms. Biohazardous waste should be properly labeled and handled according to the established protocols and regulations. Aseptic transfer is utilized in a variety of laboratory procedures, including subculturing microorganisms, transferring samples for analysis, incubating media, and preparing samples for downstream application. It is crucial in maintaining the purity and availability of microbial culture, ensuring accurate identification, characterization, and experimentation. Overall, aseptic transfer is a fundamental technique that plays a vital role in maintaining the integrity of microbial samples and culture. By following stringent practices and adhering to the principle of aseptic techniques, scientists and laboratory personnel can minimize the risk of contamination and produce reliable results in their research and diagnostic activities. Thank you for joining.